Welcome, everybody. We're very excited to host Dr. Meaghan Creed for the first JSN seminar this fall. So Dr. Creed hails from Canada. She did her PhD at the University of Toronto, where she worked with Jose Nobrega. Am I pronouncing that correctly?
It's Jose, but I always mix it up too.
Okay, Jose Nobrega. So she did a really impressive set of studies that revealed the mechanisms of deep brain stimulation, which she was using as a treatment in rodent models of dyskinesia, depression, and anxiety. And then for her postdoctoral studies, she joined the lab of Christian Luescher at the University of Geneva in Switzerland. And so there she was using insights gained from optogenetic circuit manipulations and studies of basal ganglia circuit function to develop new deep brain stimulation protocols aimed to treat addiction. So her really exciting work from there was published in Science, and she wrote a beautiful essay about these new frontiers she was developing, which earned her the inaugural Science and Pins Prize for Neuromodulation. So these are really just only a couple of the many high impact publications she has on her very impressive CV, and she's decorated with awards. As an independent investigator, she's been awarded with a Rita Allen Foundation Award for pain, a Whitehall Foundation Award. Most recently, she was awarded the Friedman Award from the Brain and Behavior Research Foundation. She's had her own lab at WashU in St. Louis since 2018, and so she's already had an incredibly productive time there. And so most recently, she published some exciting work that identified novel roles for circuits in the ventral palatum that are helping to reshape our understanding of how the basal ganglia encode value and motivation for reward, which I think she's going to talk to us about today. So please mute your microphones while Dr. Creed's talking. She welcomes people to interrupt with questions along the way, though. So if you have a question, you can either unmute yourself and just ask, or you can put it in the chat and I will monitor the chat and ask on your behalf. So please join me in welcoming Dr. Meaghan Creed.
So thank you so much, Laura, for that really generous introduction. And yep, I encourage people to stop me and ask questions. And what I'd like to tell you about today in the next hour or so, better part of an hour, is recent work from the lab that's looked at dissecting the role of these non-canonical ventral palatal populations. And towards the end, bringing this back to how we might be able to leverage these populations or this knowledge to develop new DBS-based treatments for treating disorders of reward processing. But there's no actual DBS in the talk. We're going to talk about the ventral palatum today. And to take a step back and focus on what my lab is interested in, we're really interested in the comorbidity between different disorders of reward processing. So here, this is just a really high-level overview, my synthesis of a few recent meta-analyses. We know that disorders, affective disorders, including mood disorders, chronic pain, and substance use disorders, we know that they share common genetic risk factors, and human neuroimaging studies tell us that they involve largely overlapping neural circuits. What really jumped out to me when I started looking at this was just the sheer amount of comorbidity between these conditions. So for example, for patients that are presenting with chronic pain as their primary complaint, nearly half of those patients also meet criteria for a mood disorder and over a third for a substance use disorder. Conversely, of patients presenting with a mood disorder, this was incredible. Again, a synthesis of many multiple meta-analyses, but over half of those patients meet criteria for some sort of chronic pain disorder. So these are huge odds ratios, and the extent of comorbidity between these disorders is really striking. So ultimately, what my lab is interested in is the neural circuits that mediate these core facets of reward processing that might be unique might generalize across these different disorders. And the work I'm going to tell you about today focuses really on the ventral palatum. And we're focusing on the ventral palatum for a few different reasons. But my interest in the VP really started when I was a postdoc in Christian Luescher's lab at the University of Geneva. And at the time, there was a lot of work that had been done focusing on drug-evoked synaptic plasticity in the nucleus accumbens, or the inputs to the accumbens, including the medial prefrontal cortex or the ventral tegmental area, and how addictive drugs or exposure to addictive drugs induces plasticity in this accumbal circuitry and how this leads to long-term changes in behavior. So I was interested in where that information is going. So how is that information translated beyond the accumbens, and how is that translated into the expression of drug-adaptive behavior. And I focused on the ventral pallidum, or started focusing on the ventral pallidum, because that was the key output area of the nucleus accumbens. And this work has been published for quite some time, so I'm not going to spend too much time on it. But to sum up, we actually found that in contrast to the segregated direct and indirect pathways that that have been described in the dorsal basal ganglia, that individual ventral pallidal neurons, here we're focusing on ventral pallidal neurons projecting to the VTA, but individual ventral pallidal neurons receive innervation from both D1 medium spiny neurons and D2 medium spiny neurons in the accumbens. But after high periods of activity or after exposure and withdrawal to addictive drugs, in this case, we're looking at cocaine, we saw a bidirectional occlusion of plasticity such that the D1 MSNs, their outputs to the VP got stronger, and the D2 outputs to the VP got weaker. And again, I'm not going to go into all the details of the mechanisms underlying this, but we know that it's this differential bidirectional occlusion of synaptic plasticity was related to the different complement of G protein coupled receptors and opioid neurotransmitters that they expressed. But the upshot is that following cocaine treatment, you have a strengthening and occlusion of activity-dependent plasticity in the D1 population and a weakening of plasticity or synaptic output at the D2 inputs to the ventral pallidum. And we were able to do selective optogenetic rescue of transmission at each of these pathways. So we were able to take a mouse that had undergone cocaine self-administration and stimulate the D1 output such that we could reverse or decrease the synaptic output into the ventral pallidum. We found that we could abolish or attenuate behavioral or locomotor sensitization to cocaine. And this is consistent with a large body of prior work showing the importance of the D1 MSNs in the expression of locomotor sensitization. What we showed is that it's also at the level of the output of these cells to the VP, where we can interfere and affect this behavioral sensitization. If we selectively rescued the D2 transmission by repotentiating it, we didn't see any effect on the locomotor sensitization per se, but if we looked at this amotivation or this apathy-like state that emerges after cocaine withdrawal, this is where we saw an effect. So specifically, after a mouse has undergone withdrawal from cocaine exposure, they exhibited decreased willingness to work in an effort-based task for sucrose, and they showed decreased hedonic reactions to sucrose. And we found that if we were able to repotentiate the D2 inputs to the ventral pallidum in vivo, that we could significantly attenuate some of these motivational deficits. And again, I'm not going to go into all the nitty-gritty details, but we think what's happening is that these receptors co-release opioid peptides into the ventral pallidum. We know that the accumbens is the main source of enkephalin peptides into the ventral pallidum, and that enkephalins in the ventral pallidum exert an antidepressive effect. So we think that that's the mechanism through which this is working. And I only talked about this paper to make 2 points. The first is that plasticity within the ventral pallidum was critical for the expression of both the positive sensitizing effects of cocaine exposure and was also linked to this amotivational or apathy state. So we have different types of drug adaptive behavior that were dependent on expression of plasticity at the level of the ventral pallidum. And the one thing that is a more direct segue into what I'm going to tell you about today is that when I was doing these recordings and patching all of these ventral pallidal neurons, even if we were able to narrow down our population to those neurons that project to the ventral tegmental area, there's still this huge amount of morphological heterogeneity, this huge variety of passive membrane properties in the cells we patched and their evoked firing patterns. And so one of the first goals when I started my lab was to try to dissect some of this heterogeneity within the ventral pallidum, and could we ascribe unique either neurochemical markers or biophysical properties, and could we map that onto functional differences in these ventral pallidal circuits? And so I'm going to tell you specifically about some of the work that we did characterizing glutamatergic population from the ventral pallidum, and then our more recent work looking at the so-called archepalatable neurons. So to start off, I'm just going to move my row of heads on my screen for a second. Okay. All right, so to start off, we're going to talk about the glutamatergic neurons in the ventral pallidum. And this is something that had been known really for decades, or if you look at Allen Brain Atlas, we know that VGlut2 mRNA is expressed in the VP. So this is the vesicular glutamate transporter that encodes the protein that packages glutamate into vesicles. So we know that there's mRNA in the ventral pallidum. That wasn't new, but in classical models of the VP, it's considered a homogeneous output relay station of the accumbens. So it's this inhibitory structure that receives input from the accumbens and projects downstream to areas such as the habenula and midbrain. So it wasn't clear whether these glutamatergic cells were their own unique entity or whether they represented a class of VP neurons that were inhibitory but had the capacity for glutamate co-release. And so Jessica Tooley, when she joined the lab as a research technician, this was the first year of the lab. She's now a third-year PhD student. She started to answer this question, and she did so in a pretty straightforward way using RNA-scope. And she did this with Lauren Marconi and Jason Alipio, who were a research technician and rotation student, respectively. So are these neurons their own distinct subpopulation? And so really simply, she used fluorescent in situ hybridization, where she was detecting the mRNA for this vesicular glutamate transporter, shown in red, and she was co-detecting mRNA for the vesicular GABA transporter, shown in green. And really simply, she didn't see really any overlap. I think the maximum overlap we saw was about 2% of VGAT and VGLUT overlap, which suggests that they are in fact distinct subpopulations. I'm not going to show you the data in this talk, but we also did a lot of recordings from these cells and showed that they had really unique biophysical properties as well. So now that we know that the glutamate neurons are this unique subset within an otherwise inhibitory structure, we asked how would these glutamate neurons modulate the downstream mesolimbic circuitry? And we tackled this question with patch clamp electrophysiology and circuit mapping. So we injected a Cre-dependent channelrhodopsin bilaterally into the ventral pallidum of VGLUT2 Cre mice. So we're only going to infect those VGLUT cells. And the first thing we did was just look at where we could see terminal fields. So we saw terminal fields in the classic known established projection outputs of the VP. So we saw dense labeling in the lateral habenula, in the ventral tegmental area, and in the RMTG. And if we went in and performed patch clamp recordings of these terminal fields, we found that over 50% of our target neurons that we recorded received monosynaptic excitatory currents from the ventral pallidum. So we saw this across our VTA neurons, both the dopamine neurons and the GABA neurons that we patched, as well as the habenula and the RMTG. And consistent with the fact that we didn't see much co-labeling between the VGLUT and the VGAT mRNA, we rarely saw any indication of inhibitory currents. So these are the small wimpy outward currents that you're seeing here that would reflect release of an inhibitory neurotransmitter. So we concluded that the ventral pallidum sends these monosynaptic glutamatergic inputs to these different downstream target areas. However, one of the things that we miss, especially when we prepare coronal slices and sever a lot of this network connectivity, is that these areas, the lateral habenula, VTA, RMTG, they're all highly reciprocally connected. And you can imagine that even though the VP is sending monosynaptic excitatory projections into these areas, that the actual effect or consequence of stimulating VP neurons on downstream target areas would be really highly dependent on this network connectivity. And to try to answer that question, we basically repeated the same experiment where we injected the Cre-dependent channelrhodopsin into the ventral pallidum of our VGLUT2 Cre mice. But instead of doing patch clamp recordings, we lowered acute silicone recording electrodes into each of these target areas and measured the evoked neuronal spiking in vivo in an intact neural network. And our results were a bit surprising. So in the lateral habenula, our results were what we would predict, where we had, sorry, to move your row faces again. The results are about what you would predict, where when we activated the glutamatergic neurons in the ventral pallidum, looking at the habenula spikes, we saw that of all of the units that were modulated by the light, of all the units that were significantly light modulated, they all increased their firing rate. And this was time locked to the blue light activation of the ventral pallidum. Same thing with the RMTG neurons, a little bit more diverse, but over half of the units we recorded from, these are all single units, half of those units showed significant modulation in response to BP activation. And again, most of the cells were increased their firing in response to the light. When we got to the VTA, things were a little bit more complicated, where we saw this, all of the responses were all over the place. There was a lot, surprisingly a lot fewer units that were modulated, but then we saw half that increased their firing and half that decreased their firing in response to the blue light activation. And if we scrutinize the waveforms and the properties of these single units a little bit more carefully, we found that of all the units that showed a decrease in firing rate, they all exhibited this slower triphasic waveform and a firing rate, basal firing rate below 4 hertz. Conversely, when we looked at the units that were activated, we never saw a triphasic waveform. They were all this narrower waveform, and the firing rate was more variable and typically higher, above 3 hertz. So I'm happy to talk at the end about the classical criteria people have used to distinguish, the electrophysiological criteria people use to distinguish dopamine and GABA neurons in the VTA. But what we think is happening is that through this network connectivity, that when you activate the glutamatergic ventral palatal neurons, that the dopamine neurons, consistent with this triphasic waveform, are inhibited, and conversely, that the VTA GABA neurons are excited. So this would be consistent with our patch clamp results, where if we have activation of the lateral habenula, for example, that's going to increase activity of the RMTG, which is the main inhibitory input onto the dopamine neurons. Same thing with the direct activation of the RMTG. You predict that that would also result in a dopamine neuron inhibition. So we think that by doing this in vivo intact network functional anatomy, that ultimately the effect of the stimulating the glutamatergic BP neurons is to inhibit the dopamine neurons, or that's a consequence.
Meaghan, can I ask one question about that?
Yeah.
Did you record sort of like along the axis of the VTA? Because I know there's different sort of projection defined neurons that also span the medial lateral axis. So I wonder if that could explain any of the differences as well.
Yeah, so these were all quite lateral. So we're probably avoiding, especially where we're able to even putatively say that these are consistent with the criteria for a dopamine neuron or a GABA neuron. So we're really quite in the lateral VTA. We didn't sample along the medial lateral axis. Yeah, I'd be happy to talk more about that at the end, because now that we've started doing some of the patch clamp recordings with the identified inputs, oh yeah, this criteria is definitely not valid. So I'm trying to be very cautious in my interpretation there. But I think given how lateral we were in our recordings, that we, calling them a putative dopamine neuron, I'm okay with. But yeah, we only sampled laterally. And then there was another question.
Yeah, go ahead, Priyanka.
Hello. Yeah. So my question is, how far does the VP affect the within modular connections in VTA? Is there a chance that the within network connections in VTA are so heterogeneous that we are getting these different kind of responses apart from the VTS connection with RMTG?
So I think, to rephrase the question to make sure I'm understanding, it's that we're seeing this really kind of heterogeneous population, and could this be where we're sampling, and the fact that there's many different ways that the VTA could be affected, right? It could be through direct synaptic effects or these polysynaptic effects with different latencies.
Mm-hmm. So my question was more about the connections within VTA itself. So how far, so there could be some connections which are inhibiting the VTA, other neurons in VTA. So I just want to understand the within network or the within VTA connections which might play a role in this heterogeneity.
Right. So I think that within the VTA, you have the dopamine neurons, which are projecting out of the VTA. The GABA neurons within the VTA also project out of the VTA, but there's a lot of inhibitory local connectivity. So I think that if we were, our patch clamp results basically show us that whether we're patching dopamine or GABA neurons, we see the same pattern. We see predominantly this excitatory drive, but even if you only looked within the VTA, you would expect that stimulating the VTA GABA neurons, you're going to get some inhibition of the VTA dopamine neurons. And we actually thought that that might be what we're looking at here, where we see these like really wimpy inhibitory currents in our VTA dopamine neurons, is that it's polysynaptic through the GABA neurons, but the latencies aren't consistent with that. So we think that it's, definitely through the local connectivity itself. If you're activating these VTA GABA neurons and dopamine neurons, you still might expect to ultimately get a reduction in the firing of the VTA dopamine neurons through this inhibition, this local inhibition.
Okay, thank you. Yeah.
All right, so what I've told you is that these glutamatergic neurons do seem to represent a distinct or non-canonical subpopulation, and that they can drive firing of the habenulin RMTG and ultimately inhibit what we think is putative dopamine neuron firing. And given that sort of surprising finding, we wanted to ask what the contribution of these neurons is to reward-related behavior. And we started off doing a really simple assay. So we've known for basically about 30 years that if you electrically stimulate the BP, mice will work for stimulation. They find it rewarding. We first wanted to optogenetically replicate that finding. So we injected channelrhodopsin into the ventral palatum, totally non-specifically, and we tested them in this really simple real-time place preference assay. So following the injection and time for viral expression, we also implanted light fibers directly over the BP. And we gave them a baseline test in this real-time place preference apparatus. These are two equally sized chambers with a connecting corridor. The mouse is free to move between them. We did a baseline habituation test, and then the next day we paired one compartment with optogenic stimulation of the BP. And consistent with what had been published already for electrical stimulation, we found that mice preferred to spend time on the side of the chamber where they had optogenic activation of the ventral palatum. So this is all of the neurons non-specifically. And we again found that it was at least reinforcing or rewarding that mice significantly changed their behavior to spend time on the side where they were getting stimulated. When we repeated this with just our glutamatergic neurons, we saw the opposite effect. So even though the V-glut neurons constitute about between 18 and 20% of the total population, they actively avoided spending time on the side of the chamber where they were receiving stimulation. So again, suggesting it's consistent with this idea that the predominant neuronal subtype is GABAergic, and the glutamatergic neurons might be ultimately inducing something, playing opposing roles in appetitive behavior. We also know that the ventral palatum is really critical for motivating or invigorating reward seeking. And so we wanted to look at a more sophisticated assay of reward-related behavior with our same manipulation. So we know, for example, that lesions of the ventral palatum, either electrolytic lesions, will induce a really pronounced amotivational state. It will induce animals will fail to eat or drink water. You even have some survival issues. This is based on old electrolytic lesions, and it's been replicated with non-specific inhibition of the ventral palatum, where you see a decreased vigor in reward seeking. That's a lot of work from Jocelyn Richard and Patricia Janik's group, for example. So we wanted to test the hypothesis that selectively ablating just the glutamatergic neurons might produce the opposite effect. And we did this by first training mice on a really simple fixed ratio responding task just for a single sucrose pellet. So they did pre-training where a single lever press resulted in a sucrose pellet, and we increased the lever press requirement during the training phase. And then we started baseline testing where we interspersed days of this FR5 testing, which means the mouse has to press 5 times on a lever for an individual sucrose pellet, and interspersed it with a progressive ratio task where the demand for each successive reward became higher and higher. And after the baseline training, we went in with this caspase virus, which is a Cre-dependent protein that will basically induce apoptosis selectively in those Cre-expressing cells. So we injected it to selectively ablate. This is really a sledgehammer approach to ablate those glutamatergic ventral palatal neurons, and then repeated our testing on the FR5 and progressive ratio tasks. So we found no differences in the Cre-positive mice and the wild-type littermates during the baseline training. They could distinguish the active and inactive lever. This is the number of lever presses in the task. This is the time it takes them to complete the task. And no difference in the progressive ratio, either in terms of the lever presses or the break point, which we're seeing here. And if we go in and we ablate these neurons using the caspase virus, we significantly knock down the number of glutamatergic cell bodies in the BP, and we let them recover over the next 5 days and then put them back on in their testing. And we didn't see any difference in the number of lever presses the mice were making to earn their rewards. What we did see is that they significantly reduced the time it took them to earn their 30 rewards. The sessions were capped at 30 rewards. So the mice that had the ablated V-glut neurons were earning their rewards faster, indicating that they have a higher rate of responding in the task. And then the really striking difference came when we did the progressive ratio, where we found as a group a doubling of the breakpoint only in the lesioned mice. So no change in the controls, but these mice, after we'd taken the V-glut neurons offline, as a group, they doubled their breakpoint for progressive ratio for sucrose responding. And Jess, who was the technician who ran this experiment, this was one of the very first behavioral neuroscience experiments she'd ever done. And we didn't have a timeout on the session. It was just whenever there was 20 minutes of inactivity, the session stopped. And she sent me a text after 10 p.m. at night to ask when she could go home, because this mouse had been in the box since the morning, and she'd had like brunch plans the next day. And how long was she supposed to keep doing this? That the mouse had pressed over 5,000 times for a sucrose pellet. So it was a really striking behavior result, and I think kind of spoiled her for the magnitude of effects you can expect to see in your experiments. But it did beg the question, is this, are they really just that much more motivated for sucrose, or are they insensitive to the cost associated with working for the reward? And if you combine that with this observation we had that stimulation of the neurons themselves is aversive or produces avoidance, we wanted to test this idea that the mice weren't sensitive to the consequences of the reward more explicitly. And we did this by taking mice that had already had ablations of their V-glut neurons. We let them do this just really simple free access sucrose task before pairing their sucrose consumption with a lithium chloride injection. So this is a chemical where you inject it. It induces a really profound malaise or sickness. The mice really don't like it, and it's been used to sort of devalue the sucrose reward. And again, we found something pretty striking. If you look at our control mice down here, of all the control mice, they reduced their sucrose consumption to some extent the day after we paired it with lithium chloride. But in our ablated mice, we had no change. I don't think this is a significant change, where the mice actually, some of them went back for more the next day after this really profound devaluation. And we did the appropriate motor controls, like this wasn't a stereotypy or just like an increased consumption behavior. So it really does suggest that these neurons might be playing a more unique role in constraining reward consumption. And this was shown much more elegantly by Marcus Stevenson-Jones in Bo Lee's lab more recently, where they had mice working in a head-fixed reward choice task. And I'm just going to quickly highlight the result here, is that mice had to choose to engage with the reward or not. And as the reward size increased, the rate of engagement of the mice increased. These are head-fixed, water-deprived mice. When they introduced a conflict cue, which suggested that on a proportion of trials, you're going to get an air puff with your reward, this suppressed reward responding across all sizes of reward tested. And then they went in and optogenically inhibited these glutamatergic neurons in the VP. And they also found that mice no longer updated their behavior in response to the conflict condition. So mice are behaving, even during the conflict trials, mice are behaving indistinguishably from how they behaved prior to the introduction of conflict. And so together we interpret these results to mean that these glutamatergic neurons might play a unique role in adaptively constraining reward seeking in the face of aversive consequences or reward emission. So just to summarize that these glutamatergic neurons are unique neurochemical subpopulation, that they activate these punishment centers of the brain, including the habenula and the rostromedial tegmental nucleus. And we think that they're playing an adaptive role in constraining reward seeking, especially in the face of aversive consequences. And to switch gears a little bit, I'll come back to, if we think about addiction being defined as the inability to inhibit reward-seeking behavior in the face of aversive consequences, I think it's kind of easy to think about how these, modulating these neurons might be important for treating some of the symptoms of substance use disorders. But I want to come back to that at the end of the talk and switch gears a little bit to tell you about more recent work where we've defined this distinct but also non-canonical ventral palatal population that I'm also really excited about. So as I've mentioned, the ventral palatum is classically considered, exclusively an output of the accumbens. This is according to the classical models of the basal ganglia function. Although the archepalatable neurons and archepalatable meaning distinct from the prototypic neurons that project back to midbrain and habenula. These archepalatable neurons or striatal projecting neurons have been described in the dorsal pallidus or globus pallidus. And this was really first shown by Nico Mallet working with Bolam in 2012, where he went in with a single electrode recording neurons from the GPE, juxtacellularly labeled the cells, and then post hoc reconstructed their anatomy, which is a really monumental feat. And here we're looking at a sagittal section of a reconstruction of a prototypical GPE or globus pallidus neuron, where you have the cell body in the GPE, and then it projects prototypically downstream to the entopeduncular nucleus, subthalamic nucleus, and midbrain dopamine neurons in the nigra, or midbrain neurons in the nigra. So that's the prototypical or canonical projection. He also found several examples of these, what he called the archepalatable neurons, which are, as you can see, they're projecting back up to the striatum. They have these really puny cell bodies or axons, but then they send these huge arbors that really impinge over broad striatal territories. And he's done some work showing that these are functional. These archepalatable neurons can rapidly inhibit broad territories of the striatum in vivo, and this is important for canceling action selection. So we were wondering, A, if analogous populations existed in the ventral palatum, and what the function might be. Under what conditions might you want broad inhibition of the nucleus accumbens, or when might that be adaptive? And looking at a meta-analysis from Tanisha London when she was in Lex Kravitz's lab, and she was looking at this idea of accumbal inhibition or striatal inhibition and during reward consumption. And this is a pretty robust finding. This is looking at obviously multiple different studies. And what they're looking at is across different tasks and different rewards. When you align neuronal responses in the striatum to reward consumption, what's the predominant response? And the upshot of this is that across all of these studies, across all of these different rewards, is that the primary response of striatal neurons during reward consumption is an inhibition. The mean kind of meta-analyzed summary effect is about a 72% of all the striatal neurons exhibiting an inhibition during reward consumption. So this was what we started thinking about. However, as I mentioned, these studies are all pretty heterogeneous. They're using different rewards, some natural rewards, some drug rewards, and different task designs. So some of it is cued reward seeking, some there's an effort requirement. And so we wanted to ask, under pure, unfettered conditions, whether consumption of reward was also associated with accumbal inhibition. And Jess Tooley, now as a PhD student, took this on. And what she did was, again, another simple experiment where she implanted a recording array selectively into the accumbens shell, the shell of the nucleus accumbens. And she allowed mice to do this overnight, free access sucrose drinking. So it's a dilute concentration of sucrose, and it's basically when the mouse licks on the spout, this will trigger the sucrose delivery. And that's it. That's the whole task. There's no operant requirement. There's no cues or anything like that involved. And even under these totally unconstrained conditions, what she found is that aligned to the onset of reward consumption, the predominant response was still an inhibition of accumbal units. So it was about over a third of the units we recorded from showed this inhibitory response in the accumbens shell. And then Ivan Bashez, who's a postdoc in the lab, wanted to ask, which has been proposed in the literature, that this inhibition is necessary for stopping action selection and allowing this innate behavior to be expressed. And what he did was test the sufficiency of this accumbal inhibition to promote reward consumption. And he did this by using the same free access paradigm, but this time, instead of recording the neurons from the nucleus accumbens, he applied optogenic inhibition in a closed loop manner. So he's injecting non-specific archirhodopsin, which is the inhibitory opsin, bilaterally into the accumbens shell, and the mice have optic fibers implanted over the accumbens shell. So we're ostensibly inhibiting all of the accumbal neurons in the vicinity. We're not cell type selective. And whenever the mouse licked on the spout, not only would that trigger reward delivery, but would also trigger closed loop stimulation that would only last for the duration of the lick bout. And what he found is relative to unstimulated periods where we didn't see a difference between our EYFP controls between the unstimulated and the stimulated trials. If we looked at mice that expressed the active opsin, the archepalatable, we saw a significant increase in the drinking time with this closed loop inhibition. And again, this is pretty consistent with pharmacology that's been published before, where infusion of GABA or opioids into the accumbens robustly promote reward consumption. We're just adding the time course information to that, showing that we can do this in a closed loop manner and recapitulate this enhancement of reward consumption behavior with accumbal inhibition. So that's great. We've shown you so far that this accumbal inhibition is necessary and permissive for reward consumption. And we know from prior work, don't have time to go into all of it, but really beautiful work from David Oppenheimer and Jocelyn Richard and Patricia Janik's lab, and Wayne Aldridge at University of Michigan, that the activity of ventral pallidal neurons tracks ongoing reward value on really fast timescales. And we also know, this is again from Jocelyn Richard, but also from the Monosov lab at WashU looking at non-human primates, that if you look at simultaneously reporting accumbal and ventral pallidal signals, that neural activity that's related to the value of reward predictive cue or to the reward itself, those signals can actually arise with shorter latencies in the ventral pallidum compared to the signals that are related to reward value in the nucleus accumbens. Just arguing against the idea that the information in the VP is being inherited from the accumbens. So this is, really strong functional evidence, and our hypothesis was to ask whether the ventral pallidum, especially the reward-related activity in the ventral pallidum, is one source of this inhibition to the accumbens that allows for the inhibition of accumbal units and the expression of reward consumption. And so the first thing we had to do was basically track the anatomy to ask whether the VP actually sends these functional synapses into the accumbens. And we did, similar to the story about the glutamatergic neurons I told you, we tackled this by injecting a non-specific channelrhodopsin into the VP. And we did this in mice that expressed a genetically encoded reporter protein in different subclasses of accumbal neurons, so we could know the identity of the postsynaptic cell we were recording from. So this is an example of what that looks like. So we're recording from a cholinergic interneuron that genetically encodes the TD tomato protein, and we're stimulating with blue light to activate synaptic release from these terminals from the ventral pallidum that are impinging around our recorded neurons. And this is the result of many cells, but the upshot or the takeaway message here is that if we looked at the medium spiny neurons in the accumbens, over 70% of the cells receive direct monosynaptic inhibitory inputs from the VP. And we had a smaller but significant proportion of interneurons that also receive direct inhibition from the ventral pallidum, suggesting that the VP is sending this dense, non-specific inhibitory influence into the accumbens. We also thought that there might be some regional selectivity, like areas of enrichment or de-enrichment. So this is showing the location of all of the cells we patched. And we really patched and sampled throughout the nucleus accumbens. This is showing coronal view, sort of compressed. This were the commissure. This is the midline. This is the ventral surface of the brain. And this is the distinction between the core and the shell. So we really sampled broadly throughout the accumbens shell in all of our different genotypes. And it didn't matter if we were recording from the D1 MSNs, the D2 MSNs, the parvalbumin interneurons, or the cholinergic interneurons. We really saw pretty consistent connectivity throughout the axis of the accumbens. This is just a control experiment showing that they are inhibitory currents, since they were blocked when we washed, or they were attenuated when we washed on picrotoxin, which is an antagonist of GABA receptors. But the currents that we optically evoked were not affected if we washed on antagonists of AMPA or NMDA receptors, suggesting that our glutamate population is not contributing to this archepalatable population. We're not getting any functional glutamate release out of these archepalatable neurons. And to confirm, again, as we know, the nucleus accumbens has dense connectivity. Neurons are interconnected and they're GABAergic. So again, we thought that maybe stimulating these inhibitory inputs might cause some paradoxical disinhibition, and we might get some really interesting network effects if we drove the VP terminals. But in fact, that's not what we saw. Our results were, entirely consistent with our patch data, where whether we were recording from putative medium spiny neurons or putative interneurons or multi-units, the overwhelming response was really an inhibition of the accumbal units in response to our VP terminal activation. So these are individual units shown here, and the group data shown down here. And I think we saw maybe 2 multi-units that showed some weak activation. But really the predominant response is an inhibition of the accumbens that's evoked by stimulating these archepalatable neurons. So again, we know that these archepalatal neurons inhibit the accumbens shell. And based on what I told you about the importance of this inhibition for driving reward consumption, we now asked whether the activity of these archepalatal neurons is sufficient to promote reward consumption. And the first experiment we did was just measure the endogenous activity of these neurons using fiber photometry. So we injected a retrograde Cre into the nucleus accumbens and then injected a Cre-dependent GCamp into the ventral palatum. And this is what that looks like shown here. We had a fiber coming in over the cell bodies in the VP. So we're selectively measuring the calcium signal in those archepalatal neurons during the reward consumption task. And again, this is the free access task. And what we found is that, yep, like we predict, if the archeneurons are encoding this consumption signal, we see a peak in the activity immediately after consumption onset. And we're trying to, if for anyone who's actually done any fiber photometry, you might appreciate that it's really hard to make relationships or causal inferences or correlations even between individual fiber photometry trials and behavior on any one trial. There's a lot of variability and bleaching artifacts, at least that we've had to deal with, that we find can be challenging. So what we did is we took all of these trials where the mice consumed GCamp, consumed sucrose, and split them into upper and lower quartiles based only on the GCamp signal. So for each mouse, we took all of the reward trials that they engaged in during the session, and we took the trials where the area under the curve of the GCamp signal was highest. So this is the upper 25 percentile and the lowest, which is the bottom 25 percentile split on the GCamp signal. And then we predict that if the GCamp, if the archepalatal activity was higher after reward onset, we predict that these trials be associated with increased reward consumption behavior compared to these trials where the archepalatal activity was minimal. And that is what we saw. If we look at the onset of each of the lick bouts, these are again the trials that showed the highest GCamp signal. And we see that up to 10 seconds after the lick bout, that there's a 50% chance that mice are still engaged in this lick behavior. Whereas if we looked at those trials where the archepalatal signal was lowest, almost immediately after initiating a lick bout, the probability that the mouse is still engaged in this reward consumption drops to 50%. So we do see we're able to segregate our behavior based on this initial 2 seconds of calcium signal of the area under the curve of the calcium signal. We wanted to know whether this is a heuristic control, wanting to know whether this was related to reward seeking per se. So we introduced a requirement for a nose poke before the reward delivery, and we found no GCamp significant modulation of the GCamp signal in response to the active or inactive nose poke, suggesting that it is more of a consumption signal versus a reward seeking signal. And then in the final experiment, I'll show you about is we went back to our closed loop paradigm, and we tried to, and we wanted to, our hypothesis was that, if these archepalatal neurons are inhibiting the accumbens shell, if the accumbens shell is necessary, that inhibition is necessary for reward consumption, then we should be able to promote reward consumption by stimulating these archepalatal neurons. And so we did this using testing mice across several trials, and we delivered either closed loop stimulation, which was triggered by licking on this drink spout, or open loop stimulation, where we basically took the pattern of stimulations the mice had the previous closed loop session and scrambled it and played it randomly throughout their session and asked whether the reward consumption behavior changed. So relative to our baseline pretest, we found that the no stimulation conditions, there was no difference as we'd expect. So somewhat surprisingly, in our open loop stimulation, we also didn't see any significant change in behavior. But during our closed loop stimulation, we had this massive increase selectively in the channel rhodopsin expressing mice, where they spent much more time engaging in drinking behavior compared to the unstimulated baseline. And this was not driven by the mice making more visits to the reward spout. Instead, that's shown here. Instead, they were spending more time engaging on an individual trial basis. They were spending longer at the drinking spout, consuming reward during these closed loop conditions. And to show you that data another way, if we're looking at all 10,000 trials from the 20 mice tested across the conditions, looking at the stimulated condition, it really shifted the distribution of these reward bouts out towards these longer reward bouts. And we didn't see any change in the distribution as expected with our EYFP controls.
Meaghan, there's a question in the chat just about the fiber photometry analysis. It says when you compare the 25th percent highest and lowest, is this divided into individual animal sessions or cells? And were there any patterns into which responders were high or low? Or is this noise?
Yeah, so we took every individual mouse. So each, for every single mouse, we analyzed that separately into their upper 25th percentile and their lower 25th percentile. We didn't, and that's how we compared. So then the data you're seeing is the summary of all the mice, but there's no one, it's not like all the high trials came from one mouse and all the low trials came from another mouse. So that data is like split based on the individual mice, if that makes sense. These are really long sessions, so they're overnight sessions. So there wasn't a significant variability in the number. There was like less than a twofold, variation in the number of trials that each mouse received. So they had access to drinking water, and then they had this, sucrose reward spout that they could participate in. And like you'd expect, most of the mice participated in the drinking trial. So we're really looking at, I think it was, I want to say like 800, between 600 and 800 total trials that went into those quartiles. And those were pretty equally represented among each mouse. To answer your question, but yeah, so it definitely was not the case where we have a high responding mouse and a low responding mouse, and that's what's driving the difference. This was all within subject analyzed. And we have a supplement in the paper where we show that this relationship is true for each of the individual subjects. So we plot it, for mouse A, this is his upper and lower GCAMP signal, and this is their behavior. And that relationship held in all of the mice we tested. And we did the same analysis with the GFP, because there is some variability in some GFP, motion artifacts, but we didn't see any relationship with the GFP signal. Again, we're really worried about, motion artifacts, or, the fact that they're stopping. Is that what's causing this? Is it a motor signal? And the fact that they're just not moving, is that what reflects what we're picking up? And we actually looked at during the session, we had video tracking, and during trials where the mice just went over and stopped in the corner, we also didn't see any change in the calcium signal around those events. So we did try to do all these heuristic controls to make sure we were looking at what we think we're looking at.
I also had a question about how you're interpreting the fiber photometry, like on the last slide. So it stimulating doesn't affect the number of bouts, but it seems like this fiber photometry signal ramps up in advance of reward consumption and then peaks afterwards. So what do you think that ramping up is doing?
That's a good question. That's what motivated these heuristic controls and the motor controls, especially, by definition during this task, the mouse is running over and, stopping in front of the sucrose port. So that's what we thought it might be. It might be this reward approach signal or this anticipatory signal. And we don't have a conclusive answer for what that is. We know, this is what I showed you, that we don't think it's necessarily a seeking signal. I'm just going to skip a little bit ahead to show you something that's more relevant, but we also did this in a head fix task where we intraorally infused the reward. We looked at a couple different solutions. That's just shown here. So again, because we were really trying to get at, the temporal dynamics, and especially with fibrophotometry, when it's a slower calcium signal, it's really hard. And is the increase in calcium necessarily commensurate with the firing of those neurons, or could it be like the subthreshold calcium, the inputs that are coming into the VP before the actual consumption, and you're seeing accumulation of calcium out on the processes of the neurons that's giving you this ramping signal. But if we remove the reward approach component and sort of surprise the mouse by intraorally infusing these rewards, we still see the same increase, which is again consistent with the fact that it's more of a consumption signal. But I don't have a compelling explanation for why in the free access task it begins to ramp in advance, other than if it's an anticipatory signal and perhaps reflecting more of the inputs or subthreshold calcium that's happening in advance of the actual firing of the neurons.
That makes sense.
Yeah.
And then there's 2 questions in the chat, three questions. So, okay. One says, my understanding is that this VP subpopulation inhibits the accumbens shell during hedonic reward seeking. However, do you think that this may be unique to this type of reward seeking, or do you think that you might also see this inhibition when reward seeking occurs under situations of motivational conflict?
Yeah, that's a great question, and something we're actively trying to work on. The other kind of unsatisfying idea is, we do think it's unique to reward per se, at least, insofar as we've tested. That's kind of, if we infuse something aversive and the mice spit it out, we don't see an increase. So we don't think it's just a general consumption response, but you're under conditions of conflict, is this really encoding the value, and is that value of a reward, or the anticipated value of reward, being affected by, the threat of an aversive outcome? And I think that's a really good question that we don't have the answer to. Or, could there be analogous conditions under which you'd want the accumbens to be shut down that's not reward consumption, in threat responses? I know this is maybe more true in the dorsal striatum, but if you have this kind of ongoing inhibition of action selection or the revealing of innate behavior, could you also see activity of this population that could be related to encoding potential risk or defensive behaviors? So yeah, I don't have a good answer, but it's important, and we're trying to get a handle on that. And that's unfortunately that's as satisfying as I can get with an answer. That's a really good question, though.
Okay, and then 2 more quick ones. One said, in the calcium signal, it appears as a small period of activity immediately prior to lick onset. Is this a feature of transforming the curve to derive AUC? In other words, it has to be derived from a normal distribution of values, or is this some anticipatory function of the archeneurons that encodes for expectation?
I think it's the second. You're right. So we did Z-score, we debleached and Z-scored all of our trials. So we're not looking at delta F over F. We're looking at a really like a Z-score averaged across many trials. Transforming it to get the AUC. My interpretation is that it would be an anticipatory reflection of the anticipatory activity. And again, because we don't see, any artifacts of transforming, looking at the Z-score in this preparation, for example, where there's no anticipation, we just infuse it, and you immediately, upon infusion onset, see this ramping of the signal. So I would definitely say that in the other task where they start to lick after they've initiated the consumption, that it probably reflects more of an anticipation.
Okay, there's one more, but maybe I'll save this one until after you're done, and then if people need to go.
Oh, all right. So I think the last point that I was going to make is that one of the questioners already kind of beat me to it, was about whether this was just a general consumption or whether there was a component of hedonic value that was being encoded. And we tested this in 2 ways, and it was just looking at the same closed loop stimulation during sucrose consumption. We measured the oral facial reactions to sucrose, which are unconditioned stimuli that are innate, conserved across species. And we also saw that it's a messier measure, but selectively in the channelrhodopsin group, we increased the orofacial reactions to sucrose, suggesting that the palatability of the reward is increased on the stimulated versus unstimulated trials. And then we did overnight, a 2-bottle choice overnight session where they had 2 identical sippers, identical rewards. One sipper just gave the reward, the other one gave that identical reward, but was coupled with closed loop optogenic stimulation of this population. Again, a pretty variable assay, but we didn't see any significant side preference as a group in our EYFP controls. But if we looked at the channelrhodopsin mice, all but this one guy down here developed a strong preference for the stimulation paired bottle, despite the fact that it was 2 identical rewards. And breaking this down over, we didn't just happen to get lucky and these mice had a side bias, but if we looked at the session time over the 14-hour overnight session, we see that this preference for the stimulation paired bottle in the channelrhodopsin guys is progressively developing. So this is looking at the proportion that the mice are visiting the stimulation paired bottle, and they're developing a preference for the stimulation paired bottle, despite the fact that the reward contents are identical. So we do think that together this is reasonable evidence, along with our GCAM data down here, that these archepalatable neurons are encoding the value of the reward and not just a lick or motoric consumption signal. And that was it. I'll just say that what we're doing now that I'm really excited about is understanding how self-administration of opioids affects plasticity of these populations. Specifically, if we, like the question, one of the questions asked, if these neurons are involved in encoding value, how does this go awry, and how does this plasticity at this pathway or activity or encoding in this pathway change as a function of the addiction, withdrawal, and relapse cycle? And then coming back to the first part of the talk, if we think about glutamatergic neurons playing this role in constraining reward seeking, the cardinal or a hallmark feature of substance use disorders is continuing to persist in this reward intake despite aversive consequences. And could we selectively modulate or target the ventral palatal neurons, particularly the glutamate neurons, to tone down some of this maladaptive reward seeking? So I'm excited to talk about more specific future directions, maybe offline, but yeah, with that, I'll continue taking questions if there are a couple more that popped up in the chat. Great.
Okay, thanks, Meaghan. I have one question about these 2 populations. So, I mean, it seems like their outputs are non-overlapping based on what you showed, but are they talking to each other? Like, what's the relationship between these 2 populations? Do they have shared inputs? Do they synapse onto each other? Do you know anything about that?
We don't. So I don't think the archepalatable neurons make local connectivity in the VP. We've done rabies tracing onto each population. The glutamate neurons seem to get inputs from all the areas the prototypical neuron would be expected to get inputs. So PFC, accumbens, BLA, and CEA. So all the areas that are known to project to the VP seem to impinge on the glutamatergic neurons. We know that the glutamatergic neurons do project within the VP, but we haven't been able to resolve that with cell type specificity yet. And then if this is like more or less confidential, we've tried to do rabies tracing onto the archepalatal neurons, trying to figure out where this fast signal is coming from if it's not the accumbens, and we've got no good leads. We had some hypotheses, but the rabies tracing was just really sparse and difficult to interpret. And we know that it works in our hands, so it's still like a mystery as to where that information is coming from into the archepalatal neurons. But it could be that the glutamate neurons are impinging on the archepalatals for sure. We just don't have any good sense of how these archepalatal neurons fit in.
And there's a question in the chat about the GCAMP anticipatory signal. If quinine is interpreted as a stimulus with a negative valence, what might be causing the difference between those that chose to consume versus reject the quinine? Can we decode or predict the behavioral choice of accept versus rejection of aversive stimuli based on the neural activity?
We can decode it. So we actually split these we were surprised when we saw this result. We thought that we'd see, this result here, the quinine where they consumed it was surprising. 2 things that we think, these mice are like mildly water deprived to get them to participate in the task, not substantially, but they have been water fasted for a few hours before the task. This is also a super low concentration of quinine. I'll have to double check, but it's much less than what they use for like intraoral infusions and measuring aversive reactions, because we were trying to do a free access task and get the mice to consume it voluntarily. And so we really went for this low dose quinine. So on these trials where they're consuming the quinine, again, this is not some mice consumed and some mice didn't. It tended to be like the first few trials they would consume and then they would start spitting it out. And so we can definitely correlate, if we looked at the neural activity, we can predict whether the mice on that trial would have consumed the reward. But, we're splitting this post hoc based on the behavior because we're really confused by the initial results. But that's our interpretation, is that the combination of the really low dose quinine, it's not super aversive, and the mild water restriction before we start the task is, why you're still seeing this like reward bump on a small subset of the trials. And again, it's less than what you see for plain water infusion under the same conditions, and a lot lower for the sucrose infusion. And these are all randomized order. The order of the presentation was all randomized, so there's no kind of wash in or wash out effect. But from when we analyzed the behavior, these consumption quinine trials did happen earlier in the session compared to the rejection trials. But it's a good question. Yeah.
Okay, any more questions? Beautiful talk. In the tour, in one of the figures, you showed you recorded from the shell, but I don't know if they didn't project to the core or that you didn't examine that. And the reason I ask is because the Cummins score is important for ongoing reward seeking in a lot of addiction models.
Yeah, so I think they do. I think it's, I kind of talked about this in lunch with the trainees, where I don't have any great evidence for this, but I think it's probably a scenario where you have topography in the arrangement of where the archepalatable neurons project. So I think that, if you seed, in all of our tracing studies, we were only seeding our retrograde tracer in the medial shell of the accumbens for consistency. But I think if you went a little bit more laterally out in the ventral palatum, I think that those archepalatable neurons would probably project to the accumbens core. Although we don't have well quantified direct evidence for that. Okay. But yes, I would imagine they do.
Okay, so there's two more questions and then we can let everyone go. One is, do you think with the closed loop stimulation, later on in the experiments, the mice learns to have longer drinking bouts because they start liking the reward more? So there's a reward value change.
Yeah, so we were surprised that we didn't see more of a washout. We actually, within a session, we didn't see any systematic relationship between the length of time of the reward bout. And then we did we wanted to avoid this. So, when we set this for review, we were told that, typically the way these experiments are done are with periods, 10-minute epoch of stimulation on, 10 minutes off, 10 minutes on. And we thought that would be, not the correct way to do this, because there would be some learning component that would kind of bleed over into the non-stimulation epoch. So we did pretty long washouts. So the way this was run was within a session, there was no significant relationship, but those test days were like 9 test days. And then we interspersed like no stimulation, open, closed loop. So there was a pretty good, from one session to the next, I wouldn't necessarily expect much of a carryover effect. But yeah, I'm not. And then again, I'm trying to remember how systematically we looked, because that was one hypothesis, that the bouts would get longer throughout the session. I don't think we saw evidence for that. But I think a lot of this is confounded by the length of the session and the fact that mice have this natural rhythm of consumption over their dark cycle. So, between like 3 and 4 in the morning, we saw a big peak, regardless of what else was happening. So those sorts of things make it a little bit harder to interpret. But good question.
Right. We'll do the last question. Do you ever see any satiation signal when they're consuming this sucrose water?
Not that we've looked at, but it would be cool to, especially if we had like a head fix or a food, a severely water fasted and did like a water reward and looked at, does the archipalatal signal decrease over the session? That would be, oh, we did, sorry. We, in some of our fiber photometry, especially the longer recordings, we did see evidence, especially within, bouts of reward consumption, sort of within, a 1 or 2 hour block, the decrease in the magnitude of the archipalatal signal. I don't think we made a big deal about that because we're always cautious about, what does bleaching mean with fiber photometry, especially on short timescales. But I would say yes, that we did have, especially within an epoch of multiple reward bouts, we did see some evidence for decreased magnitude of the archipalatal response with the same reward. Yeah, good question.
Okay, great. So we're over the hour, so we'll let everyone go, but thank you so much. That was